morning. Our topic for today is glycolysis or glycolysis. Glycos means sweet and lysis means breakdown. It is one of the catabolic pathways of carbohydrate metabolism. The definition of glycolysis is a sequence of reactions converting glucose to pyruvate or lactate with the production of energy. Glucose is converted to pyruvate under aerobic conditions and lactate under anaerobic conditions with the liberation of energy. It is also known as Emden-Meyerhof-Parnas pathway because it was elucidated by these three scientists. The pathway occurs in the cytosol. The purpose of this pathway is the production of energy in the form of ATP, and it is also for the formation of intermediates for other metabolic pathways. The significance of the pathways is a universal pathway because it takes place in almost all the cells of the body. The end product of aerobic glycolysis is pyruvate, and the end product of anaerobic glycolysis is lactate. Formation of lactate is a major source of energy for muscles, that is under anaerobic conditions. It is an emergency energy yielding pathway in the absence of oxygen. It is a major pathway for ATP synthesis in tissues lacking mitochondria. Since this pathway occurs in the cytoplasm, the tissues lacking mitochondria like cornea, lens of the eye, erythrocytes, etc., and tissues with few mitochondria like testes, kidney, medulla, etc., these tissues utilize this pathway for its energy purposes. This is very essential for brain, which is dependent on glucose for its energy purpose. And the intermediates of this pathway are used for the synthesis of non-essential amino acids and for glycerol, which is used for fat production. There are three phases for glycolysis. Number one is the energy investment phase. Number two is the splitting up phase. And number three is the energy yielding phase. The key enzymes of glycolysis are, number one is hexokinase, number two is phosphofructokinase, and the third one is pyruvate kinase. All three are kinases. And the rate-limiting enzyme of glycolysis is phosphofructokinase, and the only reversible kinase is phosphoglycerate kinase. The pathway starts with glucose. Glucose is converted to glucose 6-phosphate with the help of the enzyme hexokinase. Glucose is phosphorylated with the utilization of ATP. One molecule of ATP is converted to ADP. Glucose 6-phosphate is then isomerized to fructose 6-phosphate with the help of the enzyme phosphohexoisomerase. Fructose 6-phosphate, again phosphorylated by phosphofructokinase. One more molecule of ATP is utilized. The 6-carbon fructose 1,6-bisphosphate is then cleaved or splitted into 3-carbon dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by the enzyme aldolase. The dihydroxyacetone phosphate is converted back to glyceraldehyde 3-phosphate with the help of the enzyme triose phosphate isomerase. Now we have two molecules of glyceraldehyde 3-phosphate. The enzyme glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. Here, NAD is converted to NADH plus H plus. From here, we get 3 molecules of ATP, that is NADH plus H plus, enters electron transport chain where we get 3 molecules of ATP. This is an example for oxidative phosphorylation. The 1,3-bisphosphoglycerate is oxidized to 3-phosphoglycerate with the help of the enzyme phosphoglycerate kinase. This is an example for a substrate level phosphorylation. ADP is converted to ATP here. The direct capture of energy from the substrate without the help of the the direct capture of energy from the substrate without the help of the electron transport chain is known as substrate level phosphorylation. And this is the first substrate level phosphorylation in glycolysis. This step is inhibited by arsenite. The 3-phosphoglycerate is then converted to 2-phosphoglycerate with phosphoglycerate mutase. 2-phosphoglycerate then converted to phosphoenol pyruvate with the help of the enzyme enolase. Fluoride acts as the inhibitor here. Fluoride inhibits the enzyme enolase. This is why when we collect blood for blood glucose estimation, we add fluoride to it. Fluoride inhibits the pathway, thereby preventing the concentration of glucose preventing the decrease of concentration of glucose in the blood. Phosphoenol pyruvate is then converted to pyruvate with the help of the enzyme pyruvate kinase. This is a second substrate level phosphorylation. First one is 1,1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. Second one is phosphoenol pyruvate is converted to pyruvate. ADP is converted to ATP. Now we are going to discuss the energetics of this pathway. Under aerobic conditions, 8 molecules of ATPs are produced. And under anaerobic conditions, 2 molecules of ATPs are produced. Under aerobic conditions, hexokinase utilizes 1 molecule of ATP, and also phosphofructokinase utilizes 1 molecule of ATP. From glyceraldehyde, the step catalyzed by glyceraldehyde 3-phosphate dehydrogenase, NADH is liberated. That NADH enters electron transport chain, where we get for 1 molecule of NADH, we get 3 molecules of ATP. So 2 into 3, we get 6 molecules of ATP. The step catalyzed by phosphoglycerate kinase liberates 2 ATP molecules, and that of pyruvate kinase 2 molecules of ATP. So totally, we get 8 molecules of ATP in aerobic glycolysis. In anaerobic glycolysis, only 2 molecules of ATP are liberated. Next is the regulation of glycolysis, hormonal regulation. The hormone insulin stimulates hexokinase, phosphofructokinase, and pyruvate kinase. All these are the key enzymes of glycolysis. While glucagon, which is antagonistic to insulin, inhibits the same. So insulin favors glycolysis, while glucagon inhibits glycolysis. Next one is the availability of substrate, that is the food we taken. Anaerobic glycolysis. Under anaerobic conditions, that is in the absence of oxygen, pyruvate is converted to lactate by the enzyme lactate dehydrogenase. Here, NADH plus H plus is converted to NAD. The NAD produced during aerobic glycolysis is utilized here. So that is why we get only 2 molecules of ATP from anaerobic glycolysis. This is the end of the glycolytic pathway. This is one of the catabolic pathways of carbohydrate metabolism. Thank you.